A very good morning. Today I am going to present the topic somatic embryogenesis. Somatic embryogenesis forms one of the major topics in plant tissue culture, and it is a very interesting topic. Somatic embryogenesis can be defined as the development of embryos from the somatic cells. It can be defined as the process of development of bipolar structure from a non-zygotic somatic cell. Here, as the term mentioned, embryogenesis, normally embryogenesis is the development of embryos from the zygotic cells. Here, zygote is formed from the fusion of male and female gametes, and from that zygote, here embryos is developed through various stages. When it comes in the somatic embryogenesis, here there is no zygotic cells, but the embryos develop from a non-zygotic cell. It should be understand that it is having, the somatic embryo is having no connections with that of the vascular. It is having no vascular connections with that of the connecting cells. Coming to the history of somatic embryogenesis, it was introduced first by Stewart in 1958, and he developed somatic embryos from the suspension culture of carrot. Again, in 1959, Reinert extracted somatic embryos from the callus cultures of carrot. In addition of the members of the Umbellifera and Solanaceae, a range of dichotomous families have produced somatic embryos. Here, it occurs more frequently in tissue cultures as an alternate organogenesis for regeneration of the whole plant. Coming to the types of non-zygotic embryos, there are three types of non-zygotic embryos, and they are somatic embryos, parthenogenic embryos, androgenic embryos. Somatic embryos are developed from the sporophytic cells other than the zygote cells. Parthenogenic cells are developed from the unfertilized eggs. Androgenic embryos were developed from the male gametophyte, that is microspore pollen grains. Here we discuss about the differences between the somatic and the zygotic embryos. Somatic embryos, as we mentioned, somatic embryos are developed from the sporophytic cells, whereas the zygotic develop from the fertilized egg cells. Somatic embryos does not contain a seed coat, whereas the zygote cells contain a seed coat. And coming to the plantlets, here the somatic embryogenous plantlets are very weak, whereas the plantlets derived from the zygotic cells are very strong. It forms only embryos and no seed formation occurs in somatic embryogenesis, whereas the zygotic cells form seeds. The propagation rate is very high in somatic embryogenesis, whereas it is less in zygotic embryos. Then no germ endosperm formation occurs in somatic embryos, but there are endosperms formed in zygotic embryos. Here, so coming to the difference between somatic embryogenesis and zygotic embryogenesis. First, in zygotic embryogenesis, here the zygote is formed by the fusion of male and female gametes. From the zygote, it is converted to globular stage, and then heart stage, torpedo stage, then quaternary stage. After that, it is get converted to a mature plantlets. In somatic embryogenesis, first it forms the explant forms callus, and from the callus the cell is converted to embryo, and then these embryos will transform from the globular stage to the heart stage, then torpedo and quaternary stage. After it becomes a mature quaternary stage, and it will have the prominent apical meristem and apical shoot meristem and root meristem. Coming to the requirement of somatic embryogenesis, here it requires, the medium requires a high carbohydrate source, that is, here sucrose is added as a carbohydrate source in the somatic embryogenic media. Then reduced nitrogen is added. Here in the form of amino acids glutamine and asparagine is added. Then coming to the growth regulators, it forms a major portion in somatic embryogenesis. And these auxins, cytokinins and abscisic acids were added as phytohormones. Iron in this somatic embryogenesis medium, iron is used in the chelated form. And the pH culture conditions, coming to the culture condition, pH is adjusted to 5 to 7.5 and the temperature is adjusted to 25 to 30 degrees Celsius. Here in the medium of somatic embryogenesis, activated charcoal is added because these activated charcoals will absorb the phenolic compounds produced by the plants in the medium and thus facilitates the growth of somatic embryos. Then comes the steps in somatic embryogenesis. First, the induction. As we told, auxin is required for the induction of somatic embryogenesis. Auxins such as 2,4-dichlorophenoxyacetic acid, naphthalene acetic acid is added as auxin in the somatic embryogenic medium, whereas cytokinin, kinetin is added and it is for the pro-embryogenic mass are formed. Then comes to the development stage. Here in development stage of somatic embryo, auxin should be removed from the culture medium for the embryonic development, as the continuous use of auxins inhibits the development of somatic embryos. Stages formed are globula, heart, torpedo, quaternary and after that germination. In the maturation stage, the complete maturation is observed with a shoot apical meristem and a root apical meristem. Often obtain repetitive embryony in somatic embryogenesis and ABA is often required for complete maturation, but in some plants there is no need of addition of abscisic acid because it already have a prominent root and shoot systems. Sharp et al. In 1980 described about the routes of somatic embryogenesis, and he described two routes, that is one direct somatic embryogenesis and indirect somatic embryogenesis. Indirect somatic embryogenesis, here the embryo is formed directly from the explant without the formation of callus. Whereas in the case of indirect somatic embryogenesis, the embryo is developed through callus formation. There are two types of embryogenic cells. They are pre-embryonic determined cells and induced embryogenic determined cells. Pre-embryonic determined cells are those cells which are committed to form the embryos and there is no need of inducing those cells. Whereas in the case of induced embryogenic determined cells, these are normal callus cells and they are induced to form the embryo, somatic embryos, by the addition of phytohormones. Then now we discuss the steps in direct somatic embryogenesis. In direct somatic embryogenesis, here we take the explant, the somatic parts, that is the explant, somatic part as the explant, and we are culturing the explant in the nutrient medium by adding different kinds of phytohormones with the high auxin, that is 2,4-D and Na, and after that embryos were developed directly from the explant without the formation of callus. That is very important in the direct somatic embryogenesis. And here embryos are formed directly in the explant. After that maturation, then it germinates to form bipolar structure and it will be transformed into a complete plantlets, and these can be hardened and can be established in the field for further development. This is Here, the direct somatic embryogenesis in digitalis trojan even is shown. Here, the explant is cultured in the culture medium with containing high auxins, and from that the embryos are developing directly from the somatic cells without the formation of callus. After that, different stages of embryos can be seen, that is globula. Here comes the torpedo shape. It is very clear the torpedo shapes. After that, it is converted to a complete plants with a root and shoot system, and they can be directly hardened and can be established to the field. This is the direct somatic embryogenesis. Coming to the steps of indirect somatic embryogenesis, here also the explants were taken and cultured in nutrient mediums containing auxins, cytokines and other hormones, and later from that callus is formed. This is the major portion in indirect somatic embryogenesis. Here the embryos are formed from the callus. Here callus are formed. From the callus the embryos are developed and it is matured and is formed into a bipolar structure with prominent root and shoot systems, and then complete plantlets formed. These plantlets were hardened and established in the field. This is the indirect somatic embryogenesis of Eunomys elatus. Here in this, the explants were inoculated in the medium and the explants form the callus, and from the callus the embryos are developing, and thus forms the globular stage, the heart-shaped, the torpedo stage, and the complete with bipolar structures. And here it is inoculated in the nutrients free, that is hormone free nutrient mediums, and from that it is hardened, and after that it is established in the field. Now we discuss about the significance of the somatic embryogenesis. Here it can be achieved, a high rate of propagations can be achieved through somatic embryogenesis, and it is meant for the conservation of certain plants which lack vegetative propagations. There are so many plants which won't propagate vegetatively. In such plants, these somatic embryogenesis is very important, and it helps to produce the disease-free plants, and it helps in the development of synthetic seed productions. Coming to the disadvantage of somatic embryogenesis, first one is very difficult to obtain. Second, it is confined to a few species, that is it is very species explant specific. Then it is inability to generate large number of normal plantlets because there is a high chance of mutation. And it includes low frequency plant productions, production of malformed embryos, incomplete embryo maturations or low plant vigor. It may create unwanted genetic variation, that is soma-clonal variations. And in the somatic embryogenic plants, it develops certain dormancy, and it is very difficult to break that dormancy. This is the references. Then decline, conclude. Thank you.